Welcome back. There's no better way to solve a problem than by preventing it in the first place. That's especially true of issues like bovine respiratory disease. During a visit to Nebraska, Cattleman to Cattleman reporter Brian Baxter learned how cutting-edge genetic technology may help prevent diseases like BRD in the future.
In the cattle industry, no disease is more costly than bovine respiratory disease, or BRD. But a USDA-funded project called the BRD CAP, or Coordinated Agricultural Project, is working toward genetic selection tools that could allow producers to choose cattle that are less susceptible to BRD.
I think it's very important. We know that BRD costs our industry millions and millions of dollars on an annual basis, and I think it's important to research it.
In the future, DNA technology will be applied to traits that we couldn't have dreamed of before. Traits that tie to the disease complex, bovine respiratory disease, would be a key element that we haven't addressed thus far from the genomic front. But our breeders have a keen interest in better characterizing that component in the genetics of their herd.
Angus Genetics Incorporated, a division of the American Angus Association, has been helping to lead the way in providing producers better genetic decision-making tools for economically important traits using genomically enhanced expected progeny differences, or EPDs.
The genomic enhanced EPDs provide fast, real-time feedback on an individual early in life. The EPDs have always been a sound selection tool. By adding that genomic piece, you can have predictions earlier in life on a calf for traits that may be difficult to measure or not expressed early in a calf's life. EPDs are the language of the industry, and so that way the breeders and users of EPDs can just go to that one source and not try to do checks and balances on phenotypic weights or a specific genomic result. We'll boil it down into one nice clean package.
To get to that genomically enhanced EPD, a DNA sample is collected and sent to the Breed Association, where the animal information is entered into the database and the DNA sample is assigned an anonymous identifier. Then it goes to a genotyping lab like GeneSeq, where the DNA is extracted from the sample. In the case of hair cards, several follicles are punched out of the hair card and placed in a tube. In the case of blood cards, a hole punch is used to punch a sample from the blood card into the tube. Once in the tube, DNA is extracted from the sample and run over an SNP chip or bead chip to obtain a genotype. This information is converted into an MBV, a molecular breeding value.
Essentially, almost every breeding association in both beef and dairy are utilizing data that is coming from our laboratories here at GeneSeq. They're using primarily data that's collected on high-density bead chips.
This information is then returned to the Breed Association, where it's combined with the performance records and pedigree information and made available to the producer in the form of a genomically enhanced EPD.
So the future of DNA testing in beef cattle breeding, I really think on the seed stock side, it's going to become standard equipment, if you will, for selling registered Angus seed stock. As we move farther out into the future and the technology becomes more affordable, perhaps we may even see that moving, and we see it already moving into the commercial sector for aiding in selection and mating decisions in commercial cattle as well.
Producers themselves play a key role in the overall genomics process because they're the ones collecting DNA, tissue, blood, or hair samples from their cattle. Getting those collections done correctly is the first critical step to having good data to make genetic progress.
We want to make sure the FTA or blood cards are dried properly. We definitely want to avoid moldy samples or samples that have been exposed to extreme conditions, temperature, extreme sunlight, those types of things.
Common problems that are encountered with blood cards are samples with blood clots, moldy cards, and cards with tattoo ink on the card. Likewise, too little blood on the cards presents a problem of insufficient sample.
One of the most common problems that we get with samples coming in the lab would be a blood card that might be overspotted. And so I think that in general, people think more is better. In the case of a blood card, we need just really a small amount of blood, about the size of a quarter, for example. Any more than that, the DNA spot may not dry out effectively enough before it might have bacteria or fungal growth on that blood, and then that would present a problem with downstream applications.
To handle the samples, our breeders collect the DNA material, whether that's blood spotted on a card or a hair sample, for example, and they ship that material to our office here. In addition, they can go into the website on their customer account and go ahead and make that order electronically. Essentially, our customer service representatives will know that that sample is due to arrive. Once we receive that DNA material here, we'll make sure that there's a barcode either pre-printed on those cards or associated with the hair sample.
Welcome back. Let's return to the American Angus Association in St. Joseph, Missouri, and Cattleman to Cattleman reporter Brian Baxter to learn more about improvements in genetic testing and how they're shaping the future.
Other key factors in creating valuable genomic data include the speed and accuracy with which data samples are delivered to and handled by the lab.
If the information comes back from the company, the genomic result, by Tuesday afternoon, then our breeders and clientele are able to see their updated genomic enhanced EPDs on Friday morning of that week. So they have that weekly real-time information provided to them.
When it comes to tackling BRD, the research will rely on large training populations of genotyped animals with BRD health records to develop prediction equations for genomically enhanced EPDs for disease resistance.
In order to measure traits like disease resistance, we would need to look at a couple of different animal populations, a susceptible animal population and a resistant animal population. And then we would be looking at variation with respect to genetic markers for each of those animals in each of those populations.
For the breeders, genomics would be a great solution to how they could better characterize their animals for difficult to measure traits. To be able to characterize their cow herd and calf crops for BRD would be an opportunity that they could provide to their future bull buyers. They could use it in their selection programs. And with that comes some challenges. In order to hone in on a genomic piece, then we have to have appropriate training populations and learn what the DNA pieces are that need to be in an applicable test that can be taken to the ranch.
Genomics can help with difficult to measure traits like disease resistance because once training populations have been developed and tests have been developed for those traits, then producers at large in the industry can use those genomic tests on animals that may not actually have the trait measured but still make genetic progress.
From our breeders, we always receive questions about genetic selection tools for disease resistance. Bovine respiratory disease is always top of mind in our industry in terms of the costs associated with it. In future work and research, we'd like to identify genetic selection tools tied to BRD and make that a valuable future piece of the tools that we offer. Is that something that'll show up tomorrow? Probably not, but I think as an industry we have to buy in as a collaborative group to try to achieve that goal.
For producers, a decision-making tool based on genomics could ultimately help fight BRD, a disease that costs the industry as much as $1 billion a year. And that tool is on the radar screen for researchers working together in the BRD CAP project. I'm Brian Baxter reporting for NCBA's Cattleman to Cattleman.
To learn more about the USDA BRD CAP program and other industry trends, visit the project's website at brdcomplex.org or log on to our website at cattlemantocattleman.org.